Hi, everyone. We'll let people trickle in and then I will introduce Dr. Rosenblum. Give it another minute and then I'll introduce Dr. Rosenblum. All right, we're almost up to our normal viewing numbers. So I'm going to go ahead and introduce Dr. Michael Rosenblum. Dr. Rosenblum is an MD PhD, professor of dermatology and the vice chair of research at the UCSF Department of Dermatology. Dr. Rosenblum received his medical degree and PhD from the Medical College of Wisconsin. Subsequently, he completed his dermatology residency training at UCSF in the prestigious physician scientist training pathway. He joined the UCSF faculty in 2012 and since that time has become one of the world's leading skin immunologists. Dr. Rosenblum is a recipient of the Burroughs Wellcome Fund Career Award for Medical Scientists, the NIH Director's New Innovator Award, the Dermatology Foundation's Charles and Deneen Stiefel Scholar Award in Autoimmune and Connective Tissue Disorders, as well as multiple NIH R01 and R21 awards. Dr. Rosenblum was named the William Montagna Lecturer by the Society for Investigative Dermatology, and he was the scientific co-founder of Delineia Bio, which was sold to Celgene in 2017. Currently, Dr. Rosenblum is the co-founder of T-Rex Bio and Citrix Bio. Sorry if I mispronounced that.
No worries.
His research focuses on the fundamental mechanisms of how immune responses are regulated in peripheral tissues, including the skin, and how this knowledge can be exploited to treat human disease. So today he will be talking about regulatory T cell responses in the skin. So Dr. Rosenblum, go ahead.
Thanks, Jillian. I really appreciate the invitation. And thanks, Bill, as well, for inviting me. It's an honor and a pleasure to speak here today. So today I'm going to talk about what we've been up to in the lab for the last couple of years. Okay, so I'm going to advance my slides here, see if I can do that. Okay. So this is the problem. As a practicing dermatologist, I see patients with chronic inflammatory conditions of the skin and autoimmune diseases, and the skin is no stranger to these diseases. And you know, there's been a change in the dogma of the way that we think about these diseases, or at least the way that I think about these diseases. So it was once thought that you develop these diseases if you had autoreactive T cells that were escaped negative selection in the thymus and then traveled out into peripheral tissues. And then once they saw their self-antigen in the skin, they, in the right environmental circumstance, could then mount an immune response against that self-antigen, and then one would develop chronic autoimmunity and chronic inflammation. But what's changed is we now know that everyone has these autoreactive cells to specific self-antigens floating around in their blood, and negative selection actually in the thymus is not actually as robust as we thought it was. And so why do we not all go on to develop autoimmune diseases? And the thinking now is that although we have all these autoreactive cells, they're constantly being regulated by other populations of cells that are self-reactive as well, called regulatory cells. And it's really a disruption in this balance that results in autoimmunity and or chronic inflammation. And one of the major tenets of our research is that if we understand how this balance is established and maintained, will help to better understand the pathogenesis of the diseases we see in the clinic. In addition, we feel that we can develop better treatment strategies for our patients by harnessing this balance and trying to manipulate it therapeutically. So by far and away, the most well-characterized regulatory cells in both mice and humans is the FOXP3 positive regulatory T cell. And now these cells have been discovered over 30 years ago. It goes to say that we know a lot about these cells. Some things that maybe are a little less talked about with respect to these cells is that they actually like to stably reside in barrier tissues, such as the skin and gut. There's actually more Tregs in the skin and gut than any other place in the human body. They do have traditional functions. They play an indispensable role in suppressing inflammation. If both mice and human beings do not have functional Tregs, they succumb quite quickly to systemic autoimmunity. And more interesting, or not more interesting, but more recently, there's been some non-traditional functions of Tregs elucidated, and that they have specialized functions in tissues in which they reside. And so in some pioneering work done by Diane Mathis and her colleagues, they were the first to basically show that Tregs that live in visceral adipose tissue play a role in lipid and glucose metabolism. And that Tregs that come into the injured muscle play a role in muscle repair. And Sasha Rudensky showed that Tregs that come into injured lung play a role in epithelial repair in the lung. So not only do these cells actually suppress autoimmunity, they also play a role in tissue damage and have alternative functions in the tissues in they reside. And one of the central questions for my laboratory over the last 10 years has been, what is the function of these cells in skin? And so I'll take you through a little bit of that journey and then end up with a new story that we're in the process of publishing. And so first, we started to characterize Tregs in mouse skin. And as you can see here, based on this flow cytometry plot, that the skin contains quite a high number of regulatory T cells if you look in the CD4 compartment. And depending on the hair follicle cycle, anywhere between 40 and 80% of the CD4s in mouse skin are regulatory T cells. They're also enriched in human skin. These are flow cytometry of punch biopsies performed on healthy human skin. And you can see that the Tregs are enriched in the skin of healthy skin compared to peripheral blood. And they all have this sort of memory phenotype, if you will. They're all CD45RO positive. We've done some grafting experiments earlier a couple of years ago where we grafted human skin on the backs of NSG mice, and we were able to ask whether Tregs or the non-Treg CD4s or T effectors had a different propensity to leave the graft after put onto the backs of mice. And what we found is that the non-Treg CD4 compartment readily leaves the skin graft and can be found circulating in the mice, in the blood and spleen, but the Tregs actually stayed put. They basically stayed within the grafted tissue and did not migrate out in these experiments. So these are a bunch of experiments that summarize to show that we've been able to show that the skin is actually home to a very large proportion of the body's Tregs based on the size of the organ and the percentage of cells that are actually in the skin, in this organ at any one time. And that Tregs in skin have a unique phenotype. They have this sort of effector memory phenotype compared to Tregs in the blood. And that the majority of Tregs actually are tissue resident cells in the steady state. They stay put and don't continuously migrate, we believe, back and forth from the skin to the skin draining lymph nodes. And so where do they localize in skin? Well, early on, we made the observation that Tregs actually really like to be around hair follicles. And so this is mouse skin. And you can see this is the autofluorescent green epidermis. This is the interfollicular dermis. And there's these hair follicles here in the dermis. And the Tregs are kind of all around the outer root sheath of these follicles. And this is just a higher magnification of a group of follicles showing that Tregs are sort of localized to the follicular epithelium. This is a little bit more advanced way of looking at this. This is confocal microscopy, and all the Tregs are in green. And you can see these green Tregs kind of all around sometimes the isthmus and infundibular region of the follicle, other time the bulge region, but really follicularly localized. There are Tregs down in the and sort of the subcutaneous tissue down here, some of them. And occasionally you see some Tregs up and around the epidermis, but they're mostly, as you can see, localized around follicles for the most part. So yeah, so this is what I was talking about here. There are some epidermally localized Tregs here, but again, most, a large subset are localized around the follicles. Okay, so I don't want to show that again. I want to move on. Okay, there we go. So this is 2-photon microscopy now for dorsal skin of a mouse, and these are the Tregs, and this is a large sort of anagen follicle here. And what you can see is that these Tregs are basically kind of, this is a 4-hour time-lapse video where we're looking at Tregs just sort of sampling the follicular epithelium. You don't see them travel too far in this 4-hour time period. They kind of stay relatively stationary and sort of in and around, sending pseudopodia out to sample around the follicular epithelium. So the same is true for human skin. We feel that we find that Tregs are localized to both the hair follicles and the hair follicle associated vasculature in human skin as well. And this is just a large outer root sheath of a hair follicle here, and these are all Tregs. And when we quantify Tregs in punch biopsy specimens from normal healthy human skin and stratify it based on site, anatomic site, we find that Tregs are more concentrated in head and neck skin versus trunk and extremity skin. And this doesn't correlate with age or gender, just correlates with site. And this happens to be sites of skin with the highest hair follicle density. So we feel like there's a real association between Tregs and hair follicles, and that's evolutionarily conserved between mice and humans. So that's a lot of characterization. We've done a lot of functional studies looking at what Tregs actually do in the skin. And we've published these results, and I don't want to talk about them today, but suffice to say, in summary, we've shown that Tregs play a major role in hair follicle cycling. They actively interact with hair follicle stem cells and facilitate hair follicle cycling. They play a role in epidermal barrier repair. So they can, this is superficial wounding of the skin, just epidermal barrier injury, and Tregs play an active role in helping to repair the epidermis. And they also play a role in fibrosis. And so Tregs actually play a role in fibroblast biology and regulating fibroblast activation. And so these are all published works that you can go check out. What I wanted to talk about today is a news story that we recently have been working on in the lab, which is related to Tregs, but the science took us in a little bit different direction that I thought was we thought was really interesting. And so it started with some work done by Tiffany Scharschmidt when she was a postdoc in my lab. And what Tiffany found was that when you looked at neonatal skin of mice, there's this influx or wave of Tregs that comes in between day 6 and day 13 of life. And so at day 6 of life, you can see in the skin, there's a very small, or 20% of the CD4s are Tregs. And then, with 7 days later, this goes up to 80%, and then slowly reaches an equilibrium over time. But this was really interesting to us because this is a very interesting time of life for mouse skin, and that this is where hair follicle development is happening. This is where the microbiome comes in through the hair follicle that's penetrating the skin. And so there's a lot of postnatal development happening in the skin at this period of time. And so we thought that maybe this wave of Tregs that comes in during this defined very important developmental window may play a role in all of the things in the skin development, if you will, and all the things the skin has to do to develop and be a functional, viable organ. And that led to some hypotheses, and the hypotheses have to do with tissue imprinting and immune imprinting in skin. And so what we hypothesized was that maybe abnormal immune development in barrier tissues during postnatal life is actually imprinted in the tissue, and it may influence disease susceptibility later in life. And specifically, neonatal Tregs, this wave of Tregs that come into the skin during this very defined neonatal time period, may actually influence tissue development and do some sort of immune imprinting in skin that would have relevance throughout life, sort of setting the set tone, or the immune set tone of the skin may happen during this sort of critical development, postnatal development window. And so the work that I'm going to talk about today is the work by a very talented MSTB student in my lab, Ian Boothby, who basically set out in his graduate project to test these hypotheses. And so the first thing Ian did was to develop a way to transiently deplete Tregs in neonatal skin. during this defined window. So to study what happens, what Tregs are doing, the first thing we do is take them away and see what changes. And so he developed a very precise way of depleting Tregs in neonatal skin. So we utilize Sasha Rudensky's FOXP3 DTR mice, and these are mice that have FOXP3 driving the diphtheria toxin receptor. So when we give them DT, we can specifically deplete Tregs. And what Ian developed was a very low dose 2-shot DT regimen, 1 shot at day 8, and then 1 shot at day 15, and then age the mice out. And what he found was this caused a really nice reduction in Tregs. So again, 80% are Tregs in the control group at this period of time, and he was able to get that down to below 20%. And what you can see after this 2-shot regimen, the Tregs rebound really quickly, and within 20 days are back to normal and stay normal throughout the life of the animal. So it's just basically reducing Tregs in this developmental window that he was able to achieve. And nicely, there was no gross abnormalities in these mice. Everyone probably knows that if you continue to administer DT and continue to deplete Tregs, mice succumb to autoimmune disease, chronic systemic inflammation. But just this 2-shot low-dose regimen given in this very precise time point after birth led to really no gross defects in the mice. They didn't lose weight. They looked fine. They grew up normally. And so that was grossly looking at the sort of the animal level. When we started to look at the tissue level, we noticed some really interesting changes that happened. And so what we found with this neonatal Treg depletion, what happened is we lost the subcutaneous fat, and these fibrous bands started to appear in the skin that had been transiently depleted of these neonatal Tregs. And what's interesting is we didn't see tons of inflammation in the epidermis. We didn't see tons of inflammation in the dermis. We didn't even see tons of inflammation around hair follicles. We basically saw these fibrous bands and these fibrocytes really getting into the fat and replacing the fat. And this is deep. This was deep to the skin. So we saw this marked paniculitis, which is an inflammation of the fat with loss of adipocytes. And this hyperplasia of an unknown stromal cell population that form these fibrous bands in the subcutaneous fat, and the epidermal and dermal architecture relatively unaffected. What's interesting is that this doesn't happen in adult mice. So if we do the same Treg depletion regimen, but actually do it in adult mice, nothing really happens. And so you see basically the fat is maintained, there's no fibrous bands, and there's really no inflammation in the epidermis and dermis. And that's really a testament to the fact that this is a very transient low-dose Treg depletion. And so what's also interesting is the stromal rearrangement resolved over time. So if you can, we age the mice out and we depleted neonatal Tregs, you can see over time, the skin actually looked histologically normal. So these fibrous bands slowly resolved over time, and you got the adipose tissue back. And so that was also quite interesting because this was a reversible process that naturally reversed when Tregs rebounded, and basically over time, it correlated with a Treg rebound. So after Ian, we talked about these results, and after neonatal Treg reduction, it looked like the skin histology returns to normal. But to test this hypothesis, we really had to try to understand, was it really normal? I mean, it looked histologically normal, but we wanted to take a deeper analysis, or more comprehensive analysis, to see whether it was actually normal. And one observation that Ian made was that it seemed like after he depleted Tregs, there was increased Th2 cells in the skin. And he saw this in one of his flow cytometry panels of mice that he had depleted Tregs and looked later out. And so he thought that if anyone's ever stained for Th2 cytokines in mice, it's very difficult to do to get really good, robust staining. And so he crossed the FOXP3 DTR mice, which are the mice that you can deplete neonatal Tregs, to the IL-5 reporter mice. And so these read out Th2 cells because every time the IL-5 gene gets turned on, it makes a red fluorescent protein. And so Ian created these mice and then did his neonatal Treg depletion and then aged the mice out, well into adulthood, 105 days after birth. And what Ian was able to confirm his suspicion was that long term, after 100 days after depleting Tregs, there was this accumulation of skin Th2 cells in skin, as shown here by the IL-5 reporter. This is gated on CD4 positive, FOXP3 negative cells. And interestingly, in the dorsal skin of C77 black 6 mice, there are no tissue resident Th2 cells. We never see these cells in the control situations, but if you neonatal Treg deplete, you readily see this population of Th2 cells that reside in skin. And this is just the kinetics showing increased Th2 cell accumulation in skin post neonatal Treg reduction that lasts long term. And what was also quite interesting was that this was it didn't affect Th1 cells or Th17 cells. It was a very Th2 specific phenomenon. And so this is IL-5 and IL-13 production. These actually maintained increased long-term after neonatal Treg reduction, but we did not see a Th2 or Th17 accumulation long-term in the skin. So we started to hone down a little bit and ask what these Th2 cells are. And we looked at cell surface. We looked at a bunch of different things, but cell surface receptor expression was one thing that we looked at. And they actually are quite unique. So if you look at sort of tissue resident markers for lymphocytes in murine skin, CD103 and CD69 are two very classic tissue resident markers. And as you can see, the CD8s and the non-Th2 T effectors express a lot of CD103, but the Th2 cells, the red5 positive cells, did not express CD103, whereas they did express CD69, as shown here, very much like other tissue resident cells. But instead of CD103, these cells actually express CD49D, which is integrin alpha 4, or also called VLA4. And what's quite interesting is these cells express a lot of CD49D, whereas the other tissue resident cells did not express CD49D. And that's just shown here. The Th2 cells express much lower 103 compared to other tissue resident immune cells in the skin, whereas they express a lot higher levels of CD49D. So what this suggested to us is maybe based on this differential integrin expression profile that the Tregs, or that the Th2 cells that are maintained after neonatal Treg depletion, maybe these Th2 cells localize to a different area of the skin based on this integrin expression profile. And that's exactly what we found. So when you look at, this is looking at skin here, ILC2s, which are red5 but CD3 negative, can be found in the dermis and epidermis, right here in the sort of the superficial skin. And then this is the dermal white adipose tissue here. And then this is the subcutaneous tissue down here. And the Th2 cells, which are red5 and CD3 positive, are in green. And what you can see is the green cells predominantly localized in the lower portions of the skin, sort of this in the subcutis, whereas the red cells, which again are the ILC2s, were pretty much localized more in the epidermis and dermis. So this differential integrin expression profile did correlate to a differential localization, and we found preferential localization of Th2 cells in the subcutis. So I'm going to give you sort of an interim summary of the results so far, and that is we found that transient reduction in neonatal Tregs results in this sort of profound derangement of the skin stromal architecture, forming these sort of fibrous bands that resolve over time. And that this transient reduction in neonatal Tregs results in the establishment and maintenance of this tissue, the long-term maintenance of these tissue-resident Th2 cells. And that these tissue-resident Th2 cells have a unique adhesion molecule expression and predominantly localized the subcutis. So the question then became, is the stromal derangement that we're observing and the establishment of this Th2 niche related in any way? And you know, were these two separate observations that happened as a result of neonatal Treg reduction, or are they in some way related? And so we did a sort of a deep dive in looking at the stromal cell populations in the skin. And so what Ian did is we took our FOXP3 DTR neonates, we depleted Tregs, and then we sorted out the stromal cell populations, and then did single cell RNA sequencing to ask, you know, what's happening in the stromal cell populations with neonatal Treg reduction. And remember, Ian's initial hypothesis was whether this reduction in Tregs in neonatal life actually changes a development of the skin. And obviously the stromal cell populations and fibroblasts in skin play a large role in this postnatal development. And so looking at the single cell data, what Ian discovered was that there was these populations of stromal cells or fibroblasts, if you will, that increased after neonatal Treg reduction, specifically what we call cluster 6, and this cluster 6-like cells. So it's basically these two populations here, as denoted here in the single cell data. And these fibroblasts had some really interesting properties. So if you look at these clusters, they express high levels of the IL-4 receptor, the alpha, the IL-13 receptor alpha, and some interesting transcription factors, Wnt2 and Nexa3. They had a very unique sort of profile, and you can see how they segregate a little differently from the rest of the fibroblast populations. And we were specifically interested in these because they expanded after neonatal Treg depletion, and they expressed receptors for TH2 cytokines. And so what Ian was able to do, he was able to adapt our single cell data into a flow cytometry panel. And for anyone that's ever done single cell work and tried to isolate cells based on cell surface marker expression from single cell data to flow data, that's no small feat. But Ian was able to do it based on a selection of markers. So CD34, Ly6A, DPP4, and CD9 were all sort of differentially expressed in the cell population. And he was able to develop a flow panel where he gated on CD45 negative cells, CD31 EBCAM negative cells, and then he took the CD34 high cells and SCAL1 high, which are shown here. And then he took the CD9 low cells because they were lower for CD9, but high for DPP4, CD26. And so he was able to isolate these populations of fibroblasts. And to validate that we had the right cells, these cells express high levels of the IL-13 receptor and the IL-4 receptor alpha, also an Nexa3 and Wnt2, to show that he was really able to at least isolate elements of this cluster of cells based on flow cytometry. And that actually opened up a lot of avenues of therapeutic or of experimental exploration for us. And so first thing he asked, these cluster 6 fibroblasts, where do they localize? Well, they localize to the subcutis. And so this is DPP4 staining or CD26. And you can see the accumulation after neonatal Treg reduction in the subcutaneous tissue, as denoted here. And this was a statistically significant increase specifically in the subcutis after neonatal Treg reduction. So that led to another hypothesis. And so the question is are these fibroblasts that are able to respond to TH2 cytokines because they express the IL-4 and IL-13 receptor, are they able to support TH2 cells? And this premise had already been established by actually a close colleague of ours, Ari Malofsky, who showed that stromal cells, stromal cell microanatomic niches for ILC2s exist in the adventitial cuffs of the lungs, and that these stromal cells secrete IL-33, and that actually maintains ILC2s in these localized areas in the lung. And the fact that the skin TH2 cells preferentially localize the subcutis suggested that they might co-localize with these stromal cells. And so we generated another hypothesis, and that is these C6 fibroblasts may create a niche that support the maintenance and activation of TH2 cells in skin. And so we stopped calling them C6 fibroblasts, and we changed we term them TH2 interacting fascial fibroblasts, because we believe that they're derived from the fascia and that they have the ability to interact with TH2 cells in this area. And so from the remainder of my talk, I'm going to call them TIFs. And that might not be the best name, but that's what we sort of term them. And it's a little bit more descriptive than just saying C6 fibroblasts. So we started to examine these fibroblasts in depth. We looked at the IL-33 reporter mice and isolated these TIFs, and we found that TIFs are the only fibroblast population in skin that are making appreciable levels of IL-33. So this is TIF expression of IL-33. This is the bulk fibroblast. So this is all the other fibroblasts that aren't TIFs, don't make appreciable levels of IL-33, and then all the other stroma are not making IL-33. So these cells are the major source of IL-33 coming from the stromal cell populations in murine back skin. And what's interesting is that these skin Th2 cells express high levels of the IL-33 receptor. So this is shown here, looking at by the RED5 reporter and ST2 expression, and you can see that these Th2 cells express high levels of the IL-33 receptor compared to other well-known IL-33 receptor expressing cells in skin. And so we asked whether these skin Th2 cells are dependent on IL-33. And so what we did is, what Ian did was he crossed the FOXP3 DTR mice into the IL-33 knockout background and depleted Tregs, and showed that if you did not have IL-33, if the whole mouse was not able to make IL-33, you saw a significant reduction in the accumulation of Th2 cells long-term. After neonatal Treg reduction, and it didn't go to zero. So IL-33 is probably not the only factor that's required to establish and maintain these cells, but clearly plays a role. And then we did the reverse experiment where we actually injected IL-33 and see if we could augment this Th2 cell accumulation, and it was a potent stimulator of these cells. So we depleted Tregs just to get Th2 cells into the skin, and then gave IL-33. And saw a nice expansion of these Th2 cells post IL-33 injection. So these cells are able to respond to IL-33, these Th2 cells, and are somewhat dependent on IL-33 for their establishment and long-term maintenance in skin. So what Ian then did was we started to work ex vivo because we were able to isolate these cells. We started to do some ex vivo experiments to ask whether TIFs could preferentially support skin Th2 cells. And so what we did is we isolated TIFs or dermal fibroblasts, so all the other fibroblasts that weren't TIFs, and co-cultured them with skin-resident Th2 cells. And these are cells that we pulled out of the skin after neonatal Treg depletion and co-cultured them together and asked whether the TIFs preferentially could support and activate Th2 cells relative to other fibroblast populations. And what we found was that if TIFs were incubated with Th2 cells, they were significantly better at just maintaining Th2 cells in these ex vivo cultures compared to the other fibroblasts. And they did it almost as well as our positive control in this situation, in these experiments, which was IL-2, IL-7, IL-33 cocktail. TIFs were also very good at inducing the proliferation of Th2 cells, as shown here. Much better than other fibroblasts, dermal fibroblasts, in inducing proliferation of these cells, and almost as good as the positive control in inducing Th2 cell proliferation. And then when we looked at activation, which is read out by the mean fluorescent intensity of IL-5 expression in these reporter cells, we found that the TIFs were very good at inducing IL-5 expression, and actually better, slightly better than the positive control. Again, IL-2, IL-7, IL-33 cocktail. So all this data suggests that TIFs actually do preferentially support, or can actually support, skin Th2 cells ex vivo. So what about the TIFs? Are they responsive to Th2 cytokines? And so they clearly express the receptors for Th2 cytokines. So what we did is we injected IL-33 or an IL-4 and IL-13 cocktail into the backs of skin of mice, and we found that we were able to mimic these this outgrowth of these fascial fibroblasts. So you can see that these are all TIFs now that are expanded in the subcutis after injection of these cytokines, which suggests that not only do these TIFs actually support Th2 cells, but the Th2 cells can secrete cytokines that actually can support the TIFs. And again, these are super physiologic levels. We injected a lot of cytokine. This is just a proof of concept that these TIFs actually can respond to Th2 cytokines. So this is our working model. And our working model is that if Tregs are present in the neonatal time period, then they sort of suppress this niche from forming, this Th2 TIF niche, such that if you knock out Tregs, you get an expansion of the TIFs. These Th2 cells are allowed to come into the skin because they're not normally present in the skin. And when they come in the skin, there's sort of an empty house there with these TIFs for them to live. And it's this TIF Th2 axis in the lower portions of the skin that sort of create this niche for Th2 cells. And we think that one of the factors that's involved in establishing and maintaining this TIF Th2 niche is IL-33, although we think other factors are involved as well. What's interesting is we can set up this Th2 niche in skin by more than just neonatal Treg depletion. So we can actually set this up if neonatal Tregs are even there. We just have to come in with a strong Th2 stimuli. So if we come in with a very strong helminth infection, we're able to establish this Th2 niche in skin. So it's not completely dependent on neonatal Tregs, although neonatal Tregs in the steady state are suppressing the formation of this axis. There's other models too. We did ovipapain. We did several other strong Th2 stimuli to show that this wasn't just an artifact of Treg depletion, that with other strong sort of Th2 stimuli are able to get this. Another, and this is getting back to Ian's hypothesis, is what are the functional consequences? It's sort of the so what experiment, if you will. So what this Th2 niche is established. Does it mean anything for the mice as they age out? Do they have increased susceptibility to Th2 diseases or any other factor that affects their lives? And then clearly we wanted to know whether this translated to humans as well, whether this was also a phenomenon seen in human skin. So first, I'm going to talk about the functional consequences. And we focused on wound healing. And the reason why we focused on wound healing was for two reasons. Number one is that this Th2 niche this Th2 niche and TIF sort of axis is pretty deep in the skin. And for most ways we think about things like atopic dermatitis, that's much more superficial. And so, and a lot of the mouse models of atopic dermatitis have caveats. And so we didn't focus on atopic derm initially. We focused on wound healing because of a seminal paper actually published a couple of years ago showing that the major fibroblasts that contribute to wound healing actually come from the fascial layer. And these fascial fibroblasts probably play a key role in wound healing. And so we thought that because these cells were deep and because these fascial cells, these fascial fibroblasts have shown to play a role in wound healing, that maybe wound healing was the best place to look to see whether this Th2 axis may play a role later in life. And so what we did was we took mice that actually had the IL-5 reporter that was driving ROSA26 DTA. So we're able to deplete these actually mice, homozygous mice actually have a depletion of Th2 cells, and heterozygous mice report Th2 cells, but they don't have a full depletion. And then we crossed them the FOXP3 DTR. So now we have mice where we can deplete Tregs in the neonatal period and then wound them. And some of the mice will have no Th2 cells because it's totally knocked out, and other mice will have a nice Th2 compartment. And we asked whether there was any influences on wound closure and wound healing kinetics. And what we found was that after wounding, if mice had their neonatal Tregs depleted, we saw a robust expansion in the Th2 cells after wounding. And that was not seen in mice that we didn't wound, that we didn't deplete neonatal Tregs. So you needed this neonatal Treg depletion to establish this Th2 axis. And then when you wounded later in life, you saw this really nice, robust increase in the Th2 cells that accumulate in the wounded skin. And that's just shown here in a pairwise analysis. And then obviously, if you knock out in mice that had deficient Th2 cells, those cells are not, they don't have the Th2 cells because they are homozygous for the IL-5 Red5, and they actually express the DTA that knocks them out. And so that was, and this is compared pre-wounding and post-wounding. And so then looking at wounding kinetics, so we did see an increase or an enhancement in wound regeneration or wound healing kinetics in mice that had their neonatal Tregs depleted. And you can see this is shown here in the blue line. It's not a huge difference. It's better seeing it here, this rate constant, that if you see in the control animals, this was the wound healing rate. And then if you depleted neonatal Tregs, these wounds healed faster. And then if you depleted neonatal Tregs but knocked out Th2 cells, you saw a reduction back to baseline, showing that this increased wound healing rate after neonatal Treg reduction was dependent on the presence of Th2 cells in skin. And we also saw TIF expansion, and this is shown here as well as in these electrofluorescent microscopy sections. You can see that CD26 is staining TIFs. You can see some of them down here in the control situation, and that they're greatly expanded actually after neonatal T reduction. And this is actually, this whole thing is the wound here. This is the wound bed. So the TIFs greatly expanded, and then the TIF expansion was actually dependent on Th2 cells, because if you depleted neonatal Tregs but actually couldn't generate Th2 cells or accumulate Th2 cells, you saw a reduction in the TIFs back to baseline. So the TIF expansion post-wounding was Th2 cell dependent. So do TIFs exist in humans? Oh, so I want to just back up to say that yes, we believe that there are functional consequences of establishing this TIF Th2 cell axis in skin, and it does immune imprint in the skin that actually affects the skin's response to different stimuli later in life. And we're in the process of now testing other stimuli other than wound healing, but this was the first one that we tested and saw a significant difference after this niche has been established. So the next question we want to answer is, does this exist in human skin? Human skin is very different than mouse skin. And so we wanted to actually determine whether these cells, this axis existed in human skin. And so, and one of the major differences actually of human skin and mouse skin is you have this retina acutis superficialis that is this fibrous layer that runs pretty superficial in human skin. And so this is sort of the deeper layers here, but this is shown here, that you have these sort of fibrous tracts that are quite actually superficial running through the fat in human skin. And so that's a key difference between human and mouse skin. And so we were interested to see maybe this pathology or TIFs actually localize a little bit more superficial in human skin compared to mouse skin. So one of the things we did is we took human skin and we did single cell RNA sequencing analysis just on the fibroblast sorted cells, and we compared them to our TIF signature and fibroblast signature that we derived from mice. And this is a lot of bioinformatics, and a lot of integrations of data sets, and a lot of work actually went into trying to see whether this happens. But what we did is we generated an integrated cluster. So this is clustering the mouse fibroblasts and the human fibroblasts and the integrated clusters together. So these are all the mouse and human fibroblasts from, again, healthy, normal skin. And when we did the cross-species integration, we found that these TIFs are indeed present. So I want you to focus up in these clusters 12 and 19 here. When you take just the mouse cells, these are where the TIF cells are. The mouse TIFs are here based on the TIF signature. And these are also what we call quasi-mouse TIFs or pseudo-TIFs. They have a lot of expression signature, but they're not the full expression signature. So they're localized up here, this sort of, and then what we found is when we looked at the human fibroblasts, they also localized up here and they had the TIF signature. Both we call them human TIF1 and human TIF2 cells. They're very similar, but they cluster a little bit differently based on some differential gene expression. So this actually really told us that these TIFs actually do indeed exist in human skin. And this was just an odds ratio for the TIF marker enrichment in these clusters of human skin. And you can see that there's a significant association between the markers that are preferential or specific to TIFs in mouse skin and their enrichment in this cluster tenon, these clusters in human skin. So they do exist in human skin. And then we started to look at a human skin disease to see whether they might be expanded. And the disease we looked at was a disease called eosinophilic fasciitis. And the reason why we looked at EF is because it most closely resembled the phenotype or the histology that we observed in the murine skin after depleting neonatal Tregs. I remember I showed you at the beginning of the talk, those fibrous bands sort of running through the subcutis. Well, that's what you see in eosinophilic fasciitis. And it's associated with eosinophils, which is a Th2-driven phenomenon. And so this is a very rare skin disease of unknown etiology. There's only about 300 cases in the literature. It presents, it's a morbid disease where it presents with skin tightening and contraction, arthritis, muscle pain, and weakness. They have some of these patients have increased eosinophils and sed rates. And then there's kind of been an increased resurgence of this disease post-checkpoint inhibition for cancer immunotherapy, there have been case reports of people developing eosinophilic fasciitis after checkpoint inhibition. And what I wanted to sort of emphasize is that one of the reasons why we wanted to look at this disease was that the histology is very similar between what we saw in our neonatal Treg depleted mice and EF. And again, these fibrous brands running through the subcutis and replacing the adipose tissue. And so we had a patient with EF that was not on therapy, new diagnosis, come into the clinic. And that patient was very awesome and willing to be a part of our study. And so we consented the patient, they volunteered their skin, and we performed single cell RNA sequencing analysis on the fibroblasts of their skin. And what we found relative to our control skin, and these TIFs were significantly, it's hard to say significant because we only had one patient, but as a proof of concept, these cells were expanded in the EF patients, in the EF patient relative to the healthy controls. This is the patient, and this is where we see these fibrous bands down the subcutaneous fat. We stained for GATA3 and CD4. We saw an increased Th2 cells localizing with these fibroblasts in the subcutaneous fibrous tissue of this patient with EF. So, you know, n equals 1, it's a single patient, but it's suggestive that these TIFs, we know they exist in human skin, and they may be playing a role in certain human skin diseases. And there are evidenced here in EF as being expanded in this one patient and associating with Th2 cells deep in the skin. This again is our working model, that there's this time window where neonatal Tregs come into the skin. They suppress this Th2 niche from forming. If they are not present for this during this developmental window, the TIFs are able, this stromal cell population we've termed TIFs, can grow out. They can interact and maintain Th2 cells, and in turn, the Th2 cells we think can maintain the TIFs. So they set up a niche that's dependent on IL-33 as well as other factors, and the establishment and maintenance of this niche actually increases the wound healing kinetics and the wound healing dynamics, if you will. And we think that this axis may be relevant to human disease by evidence in one patient with eosinophilic fasciitis, and we're investigating this in many other human diseases as we speak. And so with that, I just want to I thanked Ian, who was this was his thesis work, and other members of my lab, collaborators, my mentor, Abul, collaborators on the project, and funding sources for a lot of the work that we do. And I'm happy to take any questions now.
That was wonderful. Thank you so much. We had a question come in the chat earlier in your talk. It's from Shahanawaz Jadeja. Is the presence of Treg cells around hair follicles to maintain the immune privilege condition? So basically just, you know, are they contributing to hair follicle immune privilege?
I think we can say definitively now that yes, that is happening. Not in this work, but in another paper that we're in the process of submitting, some work done by a different postdoc in my lab, Jarish Cohn, was able just to ablate Tregs in the skin. And we believe this is the first time that this has been done, just knocking out Tregs in a specific organ and specific tissue without affecting any other Tregs in the body. And the major phenotype in that mouse was follicular inflammation, that the hair follicle had all kinds of inflammation around it. And it was quite preferential to the hair follicle. We didn't see a lot of inflammation in other places. So I think that does answer the question that yes, in the steady state, one of the roles of Tregs is to suppress parafollicular associated inflammation.
Awesome, thank you. Hadir Shakshuk is asking, is IL-33 related to itching? I guess they're thinking of like EF patients and things like that.
It's a great question. I don't study itch. And so other people like Brian Kim and Marlis Fassett are definitely studying itch. I don't know a lot about the IL-33 pathway and itch. I do know it has been associated. I know there's clinical trials going on. So I can't speak to that with any amount of credibility.
That's all right. I appreciate you for trying. Martin Alphonse is asking, what are the levels of TGF-beta in neonatal mice, and are there any changes in the levels, and presumably during development?
Yeah, that's a great question. And we haven't, TGF-beta is complicated. I think that's like, should be TGF-beta's tagline. It's complicated, but it's a good question. And actually, we just published a paper a couple weeks ago in Science Immunology, looking at the role of Tregs and TGF-beta in skin. And what it turns out is there's a lot of different, so Tregs can make TGF-beta, they can respond to TGF-beta, and they express alpha V beta 8 integrin that actually can activate TGF-beta. And so it's not always the levels of TGF-beta that are there, it's the ability of cells to be able to activate latent TGF-beta. And so it's a complicated question that has to be examined through multiple different avenues to really get to the bottom of it. And to tell you the truth, we haven't really looked at TGF-beta or activators of TGF-beta or cells that can respond to TGF-beta in the neonatal time period. We've done it mostly in adult mice in that paper. And we actually showed that, I don't want to get into it, but we actually showed that TGF-beta pathway had a paradoxical effect. It actually leads to increased inflammation in the context of epidermal barrier repair. So it was really interesting. And this had been shown by Sasha in a different context. So I'll leave it there. But long answer to a short question. And the short answer is we haven't looked, but it's complicated.
Thank you. Sean Quattro says, great talk. What are your thoughts on the role of Tregs in keloid biology?
I think it's a great question. And I've often wanted to look at Tregs and keloids because I think that there are some reports that Tregs are increased in keloids. And there are some reports I think that are decreased too. So I think it's hard to discern. There's no doubt in my mind that Tregs play a role in fibroblast biology in the skin. And, we and others have shown that, if you knock Tregs out, you're more pro-fibrotic. And that may be related to more inflammation. And we know that inflammation usually precedes fibrosis, but it also may be inflammation independent. And there may be some direct relationships between these cells, between fibroblasts and Tregs, of which we've yet to fully explore. But I think there's a there when exploring the Treg fibroblast axis in keloids. We just haven't dug deep yet, but I think people should.
Yes, I agree. I have a question. So
Sure.
I don't know if you had seen, Bill and I had collaborated before to study JAK inhibitors in mice with glio and also eosinophilic fasciitis patients and a few adult morphia patients.
Mm-hmm.
And now I'm wondering if part of the mechanism is that we were inhibiting this Th2 axis with the JAK inhibitors. So I was kind of curious what your thoughts are for like how to target this niche and target this pathway going forward.
Yeah, I think it's a great question. I think that we have to know more about these cells in humans. I mean, this is the first identification of these cells. And I think that of TIFs, at least, obviously Th2 cells have been defined, but of these TIFs, these Th2 interacting fibroblasts. And so I feel like we need to just know more about the fundamental biology of these cells. I do think that like, if you take down the cells that support Th2 cells, it might be more therapeutically amenable or therapeutically efficacious, if you will, than taking down the Th2 cells themselves. Like, I think about it this way. It's like you can take down the soldiers that are coming out of the house, but if you take down the whole house, I think it's going to have a greater effect. We know the house is made up of these fibroblast populations, TIFs being probably a major one. There may be some others as well, but we need to know what they express, what they're, we need to know more about the biology of these cells before we can actually therapeutically target and manipulate them. That being said, I think it will have potential therapeutic benefit to target that axis. And maybe the current therapies, Dupixent and things that block receptors are actually already hitting the axis. Not just the Th2 cells, but the fibroblasts that support the Th2 cells.
Yes, makes sense. That makes me think that, there must be some epigenetic regulation going on though, because if you, like you're saying, I mean, JAK inhibitors are not durable, right? And we're hoping
Right.
for more durable treatments like taking down the house. So now I'm wondering, if they already have this epigenetic memory from the Th2 axis, and then we're going to have to do something different to actually take down the house.
Yeah, and we're doing single cell right now. We're doing, I don't know if we're not doing single cell, we're doing ATAC-seq on the tips right now to understand, like, this is getting the point, is just understand that these cells in as much comprehension as possible.
Awesome. And I have to say, I admire your work, and I think that it's really great that you're doing this because there are not many good therapies for fibrotic diseases. It's like a huge unmet need, not just the skin, right? Like, there's IPF, there's scleroderma that, can affect other organs. There's like so many diseases. So I think that's really great.
Thanks, Jillian. Appreciate it.
No, and it's been a pleasure having you on Derm Talks. I'm sure everybody in lab was very, very excited to hear you give your talk. So thank you very much.
Well, thank you for having me.
I think we're out of questions.
Okay.
So thank you so much. And we look forward to seeing everybody for our Derm Talks next month. So that's it. Take care, everybody.
Thanks, everybody.